students, welcome to our science program. At the beginning, don't forget to follow our education channel on the YouTube. Today we'll start talking about the second part of lesson 1 in unit 4, which is talking about cell division. Last time we started talking about cell division in somatic cells, and the way of division in somatic cell is mitosis division. Today we will start talking about the next or the second type of division that is called meiosis. Meiosis division is in reproductive cells for forming gametes for reproduction. So let's start. Lesson 1, Part 2, meiosis or meiotic division. Okay, it's kind of cell division that occurs in reproductive cells. So meiosis division happens in reproductive cells and leads to formation of male and female gametes and transferring genetic traits from parents to their offspring. So the importance of meiosis division is number 1, forming male and female gametes and transferring genetic traits from parents to offspring. Okay. Before cell division, cell passes through phase called interphase. Of course, we knew from our previous time that interphase means warming up. In this phase, the cell prepares itself for division. So again, in interphase, cell prepares itself for division. Okay, cell prepares itself for division and important biological process happen. Amount of genetic material duplicates once during meiosis division. Okay, phases of meiosis division take place in 2 stages that are first meiotic division, second meiotic division. So here the cell make division in 2 stages. First one producing 2 cells and in the second one producing 4 cells. So let's start. First meiotic division or the first stage. Okay, aims to reduce the number of chromosomes in the cell. Now the cell or reproductive cell contains 2n or complete number of chromosomes. Now we want to reduce this number to half its number by or to produce gametes. By the way, gametes must have n or half number of chromosomes. So the first stage aims to reduce number of chromosomes. Prophase. It's the first phase. Here we call it prophase 1. Prophase 1. Okay, here in this phase, chromatin reticulum condensed and appear in form of chromosomes. Chromosomes are arranged in homologous pairs. Each pair consists of 4 chromatids. By the way, the chromosome consists of 2 chromatids, and when the 2 chromosomes get near to each other, they form a structure consist of 4 chromatids called tetrad. Each 2 homologous chromosomes in the tetrad move away from each other. Then each chromosome consists of 2 chromatids linked together by centromere. So here, similar to mitosis, but here each 2 homologous chromosomes get near to each other and forming tetrad. Spindle appears but here each spindle fiber is connected to 1 chromosome. Nuclear membrane, of course, disappears, and we are ready to metaphase 1. Metaphase 1 after prophase 1. Here, chromosomes pairs arrange at the cell equator. In the previous division, which is mitosis, chromosomes arranged at cell equator, but here chromosomes pairs arranged at the cell equator. Anaphase, anaphase 1, anaphase 1. Spindle fibers start to shrink. Every two homologous chromosomes move away from each other. Why? By shrinking spindle fibers. So, two groups of chromosomes, not chromatids, groups of complete chromosomes move to one pole of the cell. One of the two chromosomes migrate toward the cell pole and other migrate toward the other pole. So each two chromosomes near to each other, one move to one pole and the other move to the other pole by shrinking spindle fibers. Therefore, each pole contains half number of chromosomes of the parent cell. We start by complete number of chromosomes 2n, but now we're going to form two cells. Each one contains half number of chromosomes, which is n chromosomes. Telophase 1. This is the ending of the first stage of this division. Telophase 1. Here, nuclear membrane is formed again around the chromosomes at each of the cell pools, leading to formation two nuclei. So, each group of chromosomes forming nuclear membrane around them and form separated nucleus. Each nucleus contains half number of original number of chromosomes of the parent cell. Each cell contains n chromosomes. In the case of human, reproductive cell, which is the original cell, contains 2n or 46 chromosomes. At this phase, or at the end of this stage, we have here two cells. Each one contains n chromosomes or 23 chromosomes. By the way, 2n means 46, n means 23 chromosomes or half number. After telophase 1, the cell enters in the second meiotic division. This is the second stage. Second meiotic division. It aims to increase number of produced cells from the first meiotic division. By the way, the first one, its importance for reducing number of chromosomes by forming two cells, each one contains n or half number of chromosomes. But now we make another division or the second stage to increase number of cells. So here, these are the two cells. They make the division again, passing by the same phases. But here we have prophase 2, metaphase 2, anaphase 2, and telophase 2. It's similar to mitotic division. Yes, here in prophase 2, the two centrosomes move to the two poles of the cell and forming spindle fibers. Nuclear membrane disappear and nucleolus disappear in each one of them. Here we have metaphase 2, chromosomes arranged along cell equator and connected by spindle fibers. Then spindle fibers shrink in anaphase 2, leading to splitting of chromatids to the two poles of the cell. Finally, here we have four cells. Four cells, each one contain n chromosome or half number of chromosomes. So each produced cell is called gamete that contain half number of chromosomes of the parent cell. By the way, gametes must contain n or half number of chromosomes because when they combine again, forming zygote or embryo that contains complete number again of chromosome, which is 2n. Each cell of the two cells resulted from the first meiotic division is divided in a way of similar to mitotic cell division. So, in the final phase, telophase 2 of this division, we have here 4 cells are produced, and each of them contains half number of chromosomes of the parent cells. Okay, crossover phenomena. In prophase 1, we mentioned that each two homologous chromosomes get near to each other. Why? Let's see. Here we can see the homologous chromosomes. By the way, of course, you knew that chromosomes consist of DNA and protein. And protein carrying and DNA carrying genetic traits of living organism. So now we want to make a little change in genetic material. Why? To form gametes that have little change in their genetic material, to make variation in members of the same species. So here we can see these are the two homologous chromosomes get near to each other. It consists of, let's count, 1, 2, 3, and 4. Consist of 4 chromatids. So this shape is called tetrad. The inner 2 chromatids of tetrad exchange parts of them. This causing the changing the shape of chromosome, and genetic material inside it has a little change for forming gametes that contains similar genetic material but with a little variation. This is important for variation between members of the same species. So, it's the phenomena that takes place at the end of prophase 1, where some parts of the 2 inner chromatids of each tetrad are exchanged. Why? To produce new genetic arrangement. Here we can see we have new genetic arrangement in this chromosome and in this chromosome. A little change in the genetic material. Aims or importance. Crossing over phenomena responsible for variation of genetic traits between members of the same species. So again, importance of this phenomena, which is called crossing over phenomenon, causing variation in genetic traits between members of the same species. Okay, let's see now the stages or steps of meiosis division. Of course, meiosis division is happening in reproductive cells. Before starting the division, the cell make phase called interphase. Interphase is the phase in which cell prepare itself for division. So now let's start our division. By the way, it happened in 2 stages. Okay, prophase 1 in the first stage. Here we can see nuclear membrane disappeared and nucleolus disappeared, and centrosomes moved to the 2 poles of the cell and forming spindle fibers, and each 2 similar chromosomes or homologous chromosomes get near to each other, forming structure called tetrad. After that, the second phase is called metaphase 1. Chromosomes pairs arranged at cell equator and connected by spindle fibers. Then Now we got to start the third phase, which is anaphase 1. What happened in anaphase 1? Spindle fibers shrink and chromosomes move to the two poles of the cell. This is the difference between meiosis and mitosis division. In mitosis division, chromosomes arranged at cell equator and shrinking spindle fibers causing splitting of chromosomes into two groups of chromatids. But here we can see chromosomes pairs arranged at cell equator and shrinking of spindle fibers causing the formation of two groups of chromosomes at the two poles of the cell. After anaphase 1, the third phase, next phase is called telophase 1. This is the end of the first stage of the division. We can see here nuclear membrane is formed and nucleolus is formed again, and here we have two new cells. Each one of them contain n or half number of chromosomes of the parent cell. So now number of chromosomes is reduced. Okay, meiosis division has two steps. The second step started by prophase 2. Again, nuclear membrane disappeared and nucleolus disappeared, and chromosomes are formed and connected by spindle fibers of centrosome. After metaphase 2, chromosomes arranged at cell equator, and in anaphase 2, spindle fibers shrink and two groups of chromatids move to the two poles of the cell. Finally, in telophase 2, 4 cells are formed. Each one contains half number of chromosome of the mother cell. So here we have 4 cells. Each one contains n chromosome, or 23 chromosomes in the case of human. These are the gametes of male or female. Here, the 4 cells finally are formed in telophage related to cell division. Nanotechnology and cancer treatment. Okay, what's meant by cancer and tumor? Cancer occurs when the body cells are divided continually without controlling. So, the division of body cells without any control, calling cancer disease. And the mass resulted from this called tumor. So, the uncontrolled division of body cells called cancer and forming mass of cells called tumor. Okay, using nanotechnology, scientists have developed smart microscope pumps that penetrate cancer cells and exclude them from inside to treat cancer. This is a new method to treating cancer by using nanotechnology. Okay, let's see. They were used to kill the cancer cells in experiment mice. Mice suffers from cancer were able to live 300 days after this treatment. So, it's a new method for treating cancer. Other mice that didn't receive this treatment didn't live more than 43 days. So, using nanotechnology in treating cancer cells in mice, for causing them to live 300 days more than the mice that didn't receive the same treatment. The Egyptian scientist, we must be so proud, Dr. Mustafa Said, discovered a way to detect cancer cells. We can detect cancer cells by this technology that starts by loading protein. They have the ability to attach to the cancer cells, secreting with nanomolecules of gold. So, we insert protein that can attach it to the cancer cells and carrying nanomolecules or very small molecules of gold. Injection them into the patient that is suffering from cancer. What will happen? The infected cell surface and protein get intertwined with golden molecules to make it possible to monitor. So, gold attached to these cells or cancer cells, so it be easily to be monitoring. The infected cells through microscope, each cell separately. So, gold help us to see cancer cells clearly. The method of treatment is focusing laser with certain degree to the gold molecules. Now we know the place of cancer cells by determining the place of gold molecules. So we focus laser light by certain degree to the gold molecules. Then gold molecules will absorb the light and convert light energy into heat energy, which leads to burn and kill infected cells that has stuck to them. So the protein and gold molecules, stuck to cells or cancer cells by focusing laser light, gold change or convert light energy into heat energy, and heat energy causing burning of cancer cells and leaving normal cells around them healthy. Okay, now we have a comparison between the origin of spindle fibers in animal cells and in plant cells. Let's see. Here is spindle fibers in animal cells and spindle fibers in plant cells. Spindle fibers in animal cells are formed from centrosomes from the two poles of the cells. So, in animal cell, centrosomes that move to the two poles of the cell, they form spindle fibers. This is the source of spindle fibers in animal cells. But in the case of plant cells, spindle fibers are formed from condensed cytoplasm at the two poles of the cell. Of course, because plant cell has no centrosome. So, in animal cell, spindle fibers from centrosome. In plant cells, spindle fibers are formed from cytoplasm at the two poles of the cell. Okay, let's compare between mitosis and meiosis division. Let's see. Here, mitosis and meiosis division. Location. Mitosis division happen in somatic cells, which are body cells, except nerve cells because they have no centrosomes. But meiosis division happens in reproductive cells. So, mitosis in somatic cells, meiosis in reproductive cells. Okay, the importance of division. Mitosis division for growth of the living organism and compensation of damaged cells. So, our body cells or somatic cells make cell division called mitosis for growth of the body and compensation of damaged cells. But in the case of meiosis division that happen in reproductive cells, the importance is formation of gametes of male and female, and also transfer genetic traits from generation to another. So, meiosis division, formation of gametes of male and female to perform reproduction and transfer genetic traits from generation to another. Okay, resulting cells. Mitosis division forming two identical cells. Each one contains complete number of chromosomes or diploid number of chromosomes, which is 2n. Mitosis form two cells Each one contains complete number, which is 2n. But in the case of meiosis, 4 cells are formed. Each one contains half number of chromosomes of the parent cell. The number is n, or we can say haploid number, or half number of chromosomes. If this division happen in human, mitosis forming 2 cells contains each one 46 chromosomes, and meiosis forming 4 cells, each one contains half number or 23 chromosomes that are gametes. Stages. Mitosis division has 1 step including 4 stages: prophase, metaphase, anaphase, telophase. But in the case of meiosis, we have 2 phases or 2 steps. Each one consists of 4 stages. First, meiotic, prophase 1, metaphase 1, anaphase 1, and telophase 1. Now we have 2 cells. These 2 cells divide again in the second meiotic by prophase 2, metaphase 2, anaphase 2, and telophase 2. And here at the end of telophase 2 we have 4 cells. Dear students, at the end of our lesson, don't forget the main concepts of cell division and the 2 types of cell division that are mitosis and meiosis. Mitosis that happen in somatic cells for growth and compensation of damaged cells, and meiosis division that happen in reproductive cells to form gametes and transfer genetic traits from generation to another. Thank you very much, and till we meet again, goodbye.